Assalamu alaikum. On behalf of Ahsan Academy, let me welcome you to a series of presentations on cell. This is the first presentation. So, from the background of this knowledge of level of organization, let us fix our objective. In this presentation, we will try to define cell. We will list the structural components of the cell. We'll try to find out the structure and function of individual components, and we will conclude with the relation between the structure and the function. If we study the level of organization of the human body, we will find that the cells are the structural and functional unit of the living organism. Means here it is forming the structure as an unit, and also this is the center of all functions. If the energy is produced in the form of ATP, this cell is responsible for formation of the ATP. So this is the smallest portion of the living organism, that is the unit, and it is present in the living organisms only. So these are the keywords in the definition process. So this organism may comprises of a single cell, or may comprises of uncountable cells. Let us consider the world. They're having so many countries, and each country has its peculiar shape, and this may match or very closely matching with other countries, but it usually varies from shape and sizes. Cell is like that. You can study the cells. Cells actually will not be the same. It is similar, but usually varies, even very close cells. This is a country. If we study the country, we will find that it is having one boundary outside, that is the limit of the country, and it separates the country from other country or from others like sea or any other areas. So this is the boundary, and within country we will find various component areas, and one thing is the environment. So within the boundary there is environment which may match with other country or may differ grossly. So cell has similarly one boundary that separates it from the external environment or from other cells, and within the cell, the environment that is known as protoplasm. So boundary is the plasma membrane, and environment within the protoplasm, within the cell, is the protoplasm. So if we find from the outside, the cell comprises of plasma membrane, and within it is a fluid portion known as protoplasm. So within the protoplasm, if we look into it, we will find that there is nucleus, and if we take the nucleus away, rest of the part is the cytoplasm. Within the cytoplasm, if we look, we will find that the fluid portion is cytosol, and in it there are particles matter like organelles and inclusions. Within the organelles we will get some clear fluids. This is known as hyaloplasm. So these are the components of a cell. Let us see, this figure is not complete, isn't it? Some feature, the central feature of the face is absent. So we can say all components are not present. So in other words, we can say this is before completion of the face, or in other words, we can say prototype face. In the reverse condition, if we made it complete and take it away, now we should mark it as all components are present and the face is normal. So this is EU type of face. Usually we pronounce it as U type face. So this is phase and this is U phase, we can say it in summary.
Two types of cells. The main difference is that eukaryotic cells have a double membrane-bound nucleus, which contains the cell's DNA. Prokaryotic cells do not have a nucleus, only a nucleoid, which is the central, open part of the cell where the DNA is found. Eukaryotic cells also have other large, complex, membrane-bound organelles, which prokaryotic cells lack. These include mitochondria.
These are the small cells, prokaryotic cells, have cell wall outside the plasma. There is no nuclear membrane, the nuclear materials are there, and we may not get membrane-bound organelles in it. So this is known as prokaryotic cells. And eukaryotic cells in the reverse condition, and it is the more complex and human cells. So eukaryotic cell is our subject of consideration in this presentation. Before understanding the structure, let us start with the function of the cells. You see, all reactions of the metabolism of the food we are taking takes place in the cell. So we are taking the protein, carbohydrate, fat, this is burned in the cell. So this releases the energy which is required for the maintenance of the life. So this is the center of life, and all the metabolic reactions are taking place in the cell. The second condition, cell can divide. So by division, cell can do three things. It can help in reproduction, it helps in growth and repair. So the three things, repair, growth and reproduction, are the effect of cell divisions. In a different way, we can discuss the function. Cellular activity. Different cell has specific functions or activities. Like this is the muscle cell present in the muscles, and this is responsible for contraction. In other words, we can say this is thyroid gland, and it contains the glandular cells who produces macromolecules. This is the brain, and it contains neuron. This is one type of cell which is responsible for integration of the sensory and motor functions. Let us go in detail of the plasma membrane. It's a very thin boundary of the cell which is visible only by the electron microscope and can assume the position under hyperlight microscopes also. To understand the structure, we should know the chemicals present in the plasma membrane. Those are the proteins, the lipid, and lipids are of two types, phospholipids and cholesterol. The third thing is the carbohydrate. So to understand this, we should know the structure of the plasma membrane, we should know the orientation of these chemicals in the plasma membrane. Fluid mosaic model is the most accepted theory for the explanation of the orientation of the chemical substances in the plasma membrane. So you can define it as a membrane protein that's movable mosaic within the lipid bilayers. You see, let us discuss the orientations of the phospholipid molecule. Phospholipid has two component parts, a globular head, a rounded head that is hydrophilic in nature, means has the affinity to absorb the water. So all water soluble substance can cross this head, but these heads are directed outwards and inward to the periphery of the membrane. The inner side of the membrane occupied by the hydrocarbon chains of the lipid molecule. Those are hydrophobic in nature, means they don't have any affinity, and they have phobia towards the water-soluble substance. So this central part cannot be crossed by the water-soluble substances. So this lipids makes double layers. So two heads facing outward and two tails, that is the carbon chains, faces inward of the membrane. Some scattered cholesterol molecules are observed in the membrane. Here in the upper layer, this yellow molecule is indicating the cholesterol molecules. In the fluid mosaic model, the protein molecules are oriented in two ways. It may be integral protein. You can see this protein penetrates both the layers. And the second type is the peripheral protein who floats. It may float on the outer surface and can float on the inner surface. These proteins, they do not cross the whole thickness of the membrane.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane just 2 molecules thick.
Carbohydrate lies on the outer surface, and we have seen that lipid and protein is not having any chemical bondage, so protein can float as in the lipid layer. But carbohydrate makes a chemical bondage with either protein molecule or with the lipid molecule. So if it remains attached with the protein, it forms a glycoprotein, and with the lipid, it forms the glycolipid. Carbohydrate remains on the outer surface. They often produce patches. They concentrate in some areas, and this is forming the diffuse filamentous layer over the outer surface of the cell. This layer is a highly chargeable layer and works as the barrier to many chemical substances. This is also helps in identification of the cell. Observing this pairs, as an example, we can say we determine the nature of the RBC, and we can identify which type of blood group is it. This also helps in making intercellular contact. This is a diagram showing the glycocalyx, the carbohydrate molecules making pairs on the outer surface. How to draw plasma membrane easily. You see, 3D photographs is very tough to draw, so we can easily draw a flat photographs showing all the chemical substance here. You see, this is photograph showing this phospholipid head. This is the phospholipid tail, and this is the bilayer of the phospholipid. You see, this one is the cholesterol present in the lipid layer. Proteins are oriented in two forms. This is the integral protein, and this is peripheral protein. This is peripheral protein on the inner side, and another is on the outer side. Carbohydrates making bondage with the protein. This is glycoprotein and this is glycolipid. The carbohydrate is making contact with the lipid molecule. So this is a diagram of fluid mosaic model of plasma membrane structure. Everybody knows what is this. This is aquarium. Very nice. It is having wall that is not accepting any small particles going inside even, but you see one wall is accepting a very large human being. So this wall is behaving different to the different particles or animals or living things that is trying to get inside the aquarium. And this is outer environment and this is the inner environment. So this boundary wall is actually separating the outer environment from the inner environment. And if anybody interested to signal the inner habitants should knock on the surface. So keeping this conception in mind, let us try to understand the function of the plasma membrane. Here you see the boundary is not behaving same with all. It is allowing someone to get inside which is large and not allowing someone which is very small. So we can say the plasma membrane is forming the boundary wall, and this boundary wall is selectively permeable, means the larger molecule can get in if the cell is interested, and smaller molecule may be prevented in getting entry. This is known as selective permeability of the plasma membrane. By the selective permeability, the cell can make the different environment inside than the environment prevailing outside. Some chemical substance or electrolytes may be concentrated inside and not allowing them to come out by diffusion. So inner environment becomes different from the external environment because of the presence of membrane which is selectively permeable. This membrane, like I have discussed previously, is responsible for signal reception. Suppose it's a muscle cell and nerve is interested to stimulate that muscle. So nerve aims on the surface, sends signals to the surface, and this membrane accepts the signal and sends it inside. So by this way, the cell responds to the signal which is transmitted to the plasma membrane. Fourth function, you can see the animation. A particulate matter that may be living or non-living first attaches itself to the surface, then plasma membrane surrounds it and makes a vesicle. This is known as endocytic reaction or phagocytosis, and this vesicle is formed is endocytic vesicle, or more specifically we can say this is phagocytic vesicle, and if it takes fluid, this is pinocytic vesicle or pinocytosis. So this engulfing material is done by the plasma membrane present outside, so that this particulate matter cannot do any harm to the cell. This plasma membrane makes the contact with other cells and maintain the interaction between the cells. Antigenicity is another function of the plasma membrane. We have seen that glycocalyx makes a patch, and that helps in identifying the cells. So this way the body identifies the cells and immunity against the cell will prevail. This is understanding the cell. If in some conditions this system loses the normality, then immunity system fails to identify the cell and kills the cell. So antigenicity is an important function, and maintaining the normal antigenicity is required for the cell, and which is responsibility of the plasma membrane. These are the functions, main functions I should say, of plasma membrane. What is unit membrane actually? Let us discuss this membrane we have drawn. Unit membrane means the smallest part of the membrane that contains all the ingredients, means the components and their orientations. So I have shown a small area. This is containing lipid bilayer, proteins, integral protein, peripheral protein, and carbohydrates. Seeing this smallest portion, one may have misconception that protein, that is peripheral protein, lies on the external surface only, like carbohydrate. So this should not be the unit membrane. So it should be enlarged so that this peripheral protein lying on other surface is also included. So now this smallest portion has included all the chemical components and their orientations, and observing this one can explain all the typical characteristics of a membrane. So the smallest portion of the cell membrane having its all characteristics is the unit membrane. This is the unit membrane for fluid mosaic model theory explaining the plasma membrane. What are the organelles and inclusions? Let us start with the organelles. These are the permanent inhabitants of the cytoplasm. If we consider the nucleus as an organelle, then the protoplasm contains organelles. But nucleus is not usually considered as the organelle. So in the cytoplasm, we will get some particulate matter. These are the organelles. Organelles are the permanent inhabitants, and they take part or conduct the all biological reactions that's happening in the cell. So absence of one type of organelle, totally the cell will not survive. So these are known as living components of the cytoplasm. This may be membrane bound and without membrane. This can be said membranous and non-membranous. That is also correct terminologies. One example of the membrane bound are shown here is the Golgi complex or Golgi body. This is a glass of water, everybody knows. Let us see on either side. One side there is a section of the orange, and another side is a very nice mango. So section of the orange showing a very tough outer layer and a very thin inner layer of covering. This inner layer having infolding inside. On the other side, let us have a section of the mango. This is having a thin membrane outside and inside a core. Now look at this one on the left side. This is a rounded structure, everybody knows, and it is having shape, though it is not having any membrane like the previous one. If we put all the three in this fluid, you see the shape will be maintained, whether it is having very tough outer covering, thin outer covering, or no covering. So imagine this. Using this concept, you will understand, we'll try to understand the organelles. Organelles may have membrane or maybe with. Let us differentiate organelles from inclusions. Both are present in the cytoplasm of the cell. Organelles are permanent inhabitants. So they are related to the biological reactions of the cell. So they are required for the existence of the cell, and that's why it is considered as living components. This may or may not be membrane bound. On the other hand, inclusions are temporary inhabitants, and this absence of these not interfered with the life of the cell. So one type of organelle inclusions may be absent, but cell can survive. Membrane-bound organelles are mitochondria, endoplasmic reticulum, which may be rough or smooth, Golgi complex, lysosome, peroxisome, and secretory vacuoles or granules. Secretory vacuoles is made other color because this may not be present in all cells. Without membrane, ribosome, centrosome, cytoskeleton, and proteasome. Inclusions are present in the cell, maybe lipid droplets. This is fat cell with a very large lipid droplet. Glycogen droplet, you can see it is darkly stained here. Also, some pigments are also shown. Darker blackish pigments are there. So these are the inclusions. So inclusions are the temporary inhabitants of the cell, and absence does not affect the biological chain of reaction of cell. That's why it is considered as non-living components. Thank you very much for remaining with this presentation.